Hi, I'm Katie Petrie. I'm a scientist and an assistant professor at UC San Diego, and I'm one of the authors on this paper, Destabilizing Mutations Encode Non-Genetic Variation that Drives Evolutionary Innovation. We are very excited that you will be working on a new extension of this research in your lab class. You will get to carry out the next scientific steps that will help us better understand virus evolution. But before we talk in more detail about the paper, and before we talk about the contribution that you'll be able to make, I want to spend a few minutes talking about why you or anyone else might actually care about this research. What is the key question that this research is addressing, and why is it important? The key thing this research is about is evolutionary innovation. What does this mean, and what did scientists already know about evolutionary innovation before we started this research? Innovations in evolution usually refer to big changes, things like the origin of new forms and new functions. New forms usually means new shapes and new body styles, like going from a simple sponge-like ancestor that we think was the originator of all modern animals to all the diverse shapes that animals come in today. New functions are new abilities, like a bacteria that gains the ability to digest or metabolize new food sources, like this bacteria here found in a landfill, which had actually evolved the ability to digest plastic instead of its normal carbon sources like glucose. Charles Darwin himself put it this way, writing that it is impressive that from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. Darwin is known for his theory of evolution by natural selection, which describes a mechanism for how evolutionary change works. In natural selection, any population of organisms will show variations in individual organisms' appearance or abilities, and some of these variations will enable an organism to be more likely to successfully survive and reproduce, so that it will be more likely to pass those variations on to the next generation. A well-known example is the evolution of beak size in the Galapagos finches. If a random variation increased beak size, then that finch might be better able to crack open bigger seeds and nuts and could stay better fed and more likely to survive, passing that big beak trait on to later generations. It's easy to see how natural selection can optimize existing features, like a beak, to make them a good fit for their environment. But when Darwin first came up with the theory, it was harder to see how natural selection could account for evolutionary innovations, those big changes in form and function, rather than optimization of things like the size of a beak. As some of his later critics put it, the survival of the fittest does not explain the arrival of the fittest. We can't really blame Darwin for this. Prior to the early 1900s, the scientific community didn't know the molecular basis for the variation that they saw. We now know that the source of all this variation is mutation, random changes that occur when DNA is imperfectly copied by DNA polymerase during reproduction. Mutations can alter the proteins that DNA produces. We know mutations in DNA can help optimize a form or a function by making small tweaks to the shape or structure of the proteins that they encode. But can they explain the origin of brand new forms and functions? Can simple point mutations explain all the evolutionary innovations we know have occurred during the history of life on Earth? Let's go back to the example with our bacterium and its food. At the molecular level, one of the things that enables a bacteria to use a particular food source is that it has an enzyme that's capable of breaking down its food. If we zoom into the guts of our bacterial cell, it will have protein enzymes that carry out the chemical reaction of breaking down those food molecules. Enzymes are known for being really specific. They work by having just the right shape to bind their targets. Single point mutations, changes at one amino acid position, could make small changes in the protein and affect how well the enzyme binds its target, or maybe even how fast it breaks down that target once it does bind. But for the enzyme to be able to act on an entirely new target, if that new target molecule has a different shape, usually multiple mutations are required to reshape the enzyme and give it new functionality. Why is this the case? Just like us, proteins are generally not very good at multitasking. Usually their DNA sequence is translated into a specific amino acid sequence that folds into a very specific structure, which very specifically binds to a particular target. This sets up a dilemma. How can a protein with a novel, innovative function ever evolve? Does the gene have to wait around for mutation after mutation after mutation after mutation to appear to make the changes to the enzyme big enough? Does the gene have to give up its old function? How do organisms ever gain functions then, rather than just switching between alternative functions? And maybe, more importantly, what if the old function is actually essential for the organism's survival? We know that evolutionary innovations are required to explain the diversity we see in the biosphere around us, but for many years, scientists weren't sure if selection of point mutations explained all of the innovations. In the next video, we'll talk about how a different kind of mutation provided a solution to this multitasking dilemma.